This video is for The Living Earth, Unit 2.2, Photosynthesis. Today's essential question: how does the process of photosynthesis work? Our first main idea: light-dependent reactions. The overall biochemical reaction for photosynthesis is a very complex process. This complex process can be divided into two main stages: the light-dependent reaction and the light-independent reaction, which is also known as the Calvin cycle. But these stages take place inside the chloroplast. Both stages take place inside the chloroplast. The light-dependent reaction, or in the light-dependent reaction, solar energy is converted into potential chemical energy. The light-dependent reaction takes place specifically in the thylakoid, which is a stacked sac-like structure found within the chloroplast of plant cells. These structures contain the green pigment called chlorophyll. Chlorophyll absorbs wavelengths of light other than green. Other different colored pigments may also be present and help to absorb light. However, chlorophyll is the most important of all the different pigments for photosynthesis. The light-dependent reaction has two photosystems, referred to as photosystem 2 and photosystem 1. And the light-dependent reaction takes place in the following steps. 1. Photosystem 2 occurs first. I know that seems funny, but in photosystem 2, light is absorbed by chlorophyll molecules that excites electrons in the chlorophyll. These excited electrons are passed onto a series of proteins called an electron transport chain. Stage number 2. Electrons are passed along this electron transport chain, and at each link, the electrons lose some of the energy they gained from the light. This energy is captured and used to convert ADP, which is adenosine diphosphate, into ATP, which is adenosine triphosphate. This process is called photophosphorylation. Water is split in this process. Hydrogen ions and oxygen are produced. Oxygen, which is from the water, is given off as a byproduct. Stage number 3. In photosystem 1, the same electrons are re-energized by even more light and pass down a second electron transport chain. Stage number 4. The electrons passed down the second electron transport chain are used by the hydrogen ions, to convert NADP into NADPH. Stage number 5. The potential difference created by the electron transport chain, resulting in a differing number of hydrogen ions on either side of the cell membrane, causes transport of a hydrogen ion down the gradient, which produces energy for phosphorylation of ADP into ATP, which is mediated by an enzyme known as ATP synthase. So notice in this representation of the inner space of a thylakoid that in this system, photosystem 2 and photosystem 1, notice that light in the form of photons is used twice in this process. First, light is used in photosystem 2 to initially excite the electrons and to make ATP and then split water. Then, light is used in photosystem 1 to re-energize the electrons to make NADPH. So, in the light-dependent reaction, light energy has been converted into chemical potential energy in high-energy molecules like ATP and NADPH, which again are both sources of high-energy electrons. These high-energy molecules are passed on to the next stage of photosynthesis, the light-independent reaction. Our next main idea, the light-independent reaction, also known as the Calvin cycle. The Calvin cycle takes place in the stroma of the chloroplast. It uses carbon dioxide to make organic molecules that are then converted into sugars. No light is used in this part of photosynthesis because it is powered by ATP and NADPH from the previous light reaction. Now, the steps are, step number 1, carbon dioxide from the air is captured by existing organic compounds, or CO2 acceptors. This process is called carbon fixation. Step number 2, the carbon from the carbon dioxide is then reduced through a series of steps by electrons provided by the NADPH, which was produced in the light reaction. Stage number 3, with help from the energy provided by ATP, which again came from the light-dependent reaction, a sugar-like compound containing 3 carbons is made. Some of this 3-carbon substance exit the Calvin cycle and is used to make glucose. Now, in the light-independent reaction, carbon from carbon dioxide is fixed into organic molecules that are then turned into sugars. These can be used to make other organic molecules in addition. These compounds may be used by the plant directly or passed down the food chain.
The light-independent reactions of photosynthesis, also known as the Calvin cycle, can take place both in sunlight or in darkness and result in the creation of energy-rich sugars. These reactions occur in the fluid-filled stroma of the chloroplasts and require carbon dioxide from the air, as well as the ATP and NADPH produced during the light-dependent reactions of photosynthesis. The Calvin cycle begins with the critical process of carbon fixation, which occurs when a carbon dioxide molecule is added to a CO2 acceptor molecule known as ribulose biphosphate, or RUBP, which contains 5 carbon atoms and 2 phosphate groups. This reaction, which only occurs in the presence of an enzyme known as rubisco, produces an unstable 6-carbon product that immediately splits into 2 identical 3-carbon sugar compounds known as 3 PGAs. After this occurs, one of the 3 PGA molecules exits the cycle and will be used to make glucose and other sugars. The other molecule remains in the cycle. First, it reacts with ATP. Then, the product of this reaction receives a hydrogen atom from NADPH, producing NADP and a new carbohydrate compound known as G3P. After that, a complex series of steps known as the regeneration of ribulose takes place that consumes another molecule of ATP. These steps result in the formation of a new ribulose biphosphate molecule, and with that, the Calvin cycle is complete. Now the cycle repeats itself with the fixation of a second molecule of CO2. Note that 1 repetition of the Calvin cycle is required for each atom of carbon that is added or fixed to a carbohydrate molecule. So in order to make 1 molecule of 3-PGA, which contains 3 atoms of carbon, 3 repetitions of the cycle are required. That means the Calvin cycle must be repeated 6 times to make 1 6-carbon glucose molecule from 2 3-PGA molecules. Glucose is the ultimate product of photosynthesis because its chemical bonds store the energy that was captured from sunlight during the light reactions of photosynthesis. And glucose is the key nutrient that is broken down during aerobic respiration to generate the ATP that is used to power the activities of living things.